Recordings are very costly. And to pay per GB, in dollars with tax.
And then instead you can store in your laptop, I think so.
No, actually, where I had to upload. That is the thing. Because now after that, again, I had to upload here only. Mm. Either I had to upload in somewhere. No, it is all technology. We are all bind in a loop, actually. We're already in the loop, actually. It's difficult to get away from that. Whether any platform, it is very costly, actually. So that's why I'm charging. Otherwise, I had to teach in YouTube only. But it's two-way interaction is not possible there. Then, actually, tax is very, very high. Even the trolls, I say, okay, some why they are trolling. When you pay, then only you'll get. Tax is very high. And we discussed about MABB yesterday. What are the different schemes? Like, what are the different steps of MABB? Can anyone tell?
Foreground selection, recombinant selection, background selection.
Foreground, background, and recombinant.
Recommend.
Foreground, background, recombinant. Okay, we have discussed thoroughly. I will not take again. Okay, I think many of you clear like what is MABB, what is the time required, what is reliability of the selection, what is the selection efficiency, how we, if the what is the prerequisite MA for MABB, what is the advantages over the conventional breeding. See, you should read in all aspects. If you're reading on topic means you should cover in the all aspects. May I am also missing some points here because I can't accommodate everything. There are several points in the books, and different books will cover differently. And for example, there is one of the variety PJMJ. Who will know this variety and who will read this? And they will directly ask which is the variety of it is variety of which crop. Who will know? Those who are working in these crops, they will know. And the paper is also so much biased. See, if the wheat breeder and maize breeder will prepare the paper, they'll put all the questions from wheat and maize. Whatever the code. For example, you take 4 EMS system. It is present only in the wheat. Either you take haploid, like stock 6. It is only in the maize. And Agilops ventricosa. It's a landrace. Actually, I think it's some species, not landrace. Either it's one of the species of wheat, uh, it is belongs to wheat. Question came. And biscuit making variety of wheat, of hull. Who will read all those things from? Okay, they are very important for that particular crop. Those who are working in the wheat or maize, they used to write thesis, they used to collect review of literature. Some other the way they will collect the information. They will get the answers. But see, I am working in rice. Hardly 1 hour, at least no questions from rice also. And those who are working in now, I am working in pulse. Pulse, no questions. Like this, there will be paper is so biased actually. And what are the things like even take atrocious breeding related, they will be in major questions. Or like several other questions also there. But other questions are known by everyone. But these specific questions which are not known to everyone. That's why SRF, like those who are working in the crop, they get benefit and they will get good ranks. This is what actually happening. So that's why reading in every crop is very important. 80% your chance, but 20% is luck to getting to IRA. Okay. I don't know. Next year, who will prepare? Next year, if the rice breeder will prepare the questions, maybe we'll get most of the questions which are related to rice. You can observe the questions neatly. You can get if you analyze the question paper, you will get to know. And most of the related to cross-pollinated and like everywhere around the like only their specific areas. Those who are preparing papers specific to their areas only the questions. Really, it's not that a good approach to prepare the papers. But in ASRB, it is not like that. You can get question from every part and every line in the BDSing is very important. 150 questions, I think. 150 different topics will be covered. All the topics will be covered. And questions are very easy, one-liner. That is another disadvantage. But okay. For example, you are working in rice, like if you are working in a wheat crop, whatever it may be the research topic, at least you should know about, at least you are knowing about the species, what are different evolution pattern, what are the important varieties, what advancement is happening in the wheat, specific to wheat, cytogenetic approach, all the things you are getting. But those who are working in any other crop, what is the necessary to read? Okay, getting knowing the things is good, but I think that there are millions of topics in the genetics, the conceptual. Well, we'll leave that, but they're specific to that. Okay, don't worry. These are the things. It's always there in every exam. That's why reading in every aspect is very, very important. Then, today we'll just going to discuss about marker restricted recurrent selection. We'll go to directly scheme. What is recurrent selection? Can anyone tell what is recurrent selection? What are the types? Who discovered it? Recurrent selection.
It's a population improvement method used for improving alleles in the population, for aggregating positive alleles in the population.
You told the objectives. What exactly recurrent selection? The definition of recurrent selection. See, this
Continuous
is also
cycle
another
of
mistake what we will read all the things which are related to that, but we'll forget what exactly it is.
continuous cycle of selection and crossing.
Yeah, this should become selection after selection with intermating. Then again we'll go selection, selection, selfing, intercross, again select, intercross. The cycle will go on. Okay, this is actually the definition. Can you repeat again?
Sir, generation after generation, we are going to select and cross.
Yeah. Why we need to generate? See, in the name only, recurrent means again and again. Recurrence, okay? Again and again, we are doing selection. Again and why you are doing again and again? After every selection, you are intercross the selected individual. Again, variability is created and you are selecting upon the variability again. Then again you are going to cross. Again, you create intermediate, like sorry, heterozygous populations. Okay, this is actually the definition. Why we are doing it? Why specifically we have to select self cross? Why it is
Sir,
happening?
we have to increase
Two major
uh,
objectives for that.
we have to increase the positive values. Meanwhile, we have to maintain the diversity.
Yeah, not diversity. It should maintain the
This is
heterogeneity in
heterozygous.
the population. Because I told because of the cross. Mostly these approaches followed in cross-pollinated crops. Means because of heterozygous balance, you should keep the population in heterozygous state only. If you keep them in a homozygous state, there will be a chance of inbreeding depression. Okay? That's why one of the main objectives, one of the two objectives, there one is maintain the heterogeneity in the population to maintain heterozygous condition. Another objective is You already told the
Aggregating the
answer.
positive.
Increase.
Aggregating
Combining.
the positive alleles in the population. Aggregating the positive alleles in the population. This is the two very important. And see another question I remember. See, actually, how it biased paper. LR34 was a gene. Okay, reading about remembering. Okay, the registration gene is okay, but it is only first and the genetic mechanism of LR34, ABC transporter or this. Who will know? Except those who are working in the rice and that to specific labs working only on the rust, they are knowing these things. How other people can know this? But reading is good, but this is the thing. Okay, should be in generalized. Yeah. Then recurrent selection. Then one is to maintain the heterozygous condition, and another is to maintain the aggregating the positive alleles or positive genes in the population. This is the two important objective you should remember, and you should remember the definition of recurrent selection and two objectives of recurrent selection. You will get full marks. If you wrote all the things, okay, aggregating population, heterozygous, then question will come, how will do it? So that is the thing. Okay, when you're writing the answer, you should cover everything. Then, what is different schemes of recurrent selection?
Simple recurrent selection by GCA, SCA,
Who
reciprocal
given the
recurrent
recurrent
selection.
selection? CA, recurrent selection. CA. I will deal again. Okay, don't worry. These things I will deal again when I will dealing with heterosis breeding. Recurrent selection for SCA, recurrent selection GCA, and reciprocal recurrent selection. At least you should remember the scientist and what are the different schemes. Okay, and what happens in GCA, what happens in SCA, what happens in simple recurrent selection, what happens in recurrent selection. Should know all the things. All the objective is to improve this only, to maintain heterozygosity and it also maintain the variable, as I told, variability in the population, so that you can have a more chance to select the better individuals. Okay, and to aggregate all the positive alleles from the different parents into a population, and you can improve the population as a whole. Here in this one, in any breeding method in cross-pollinated crops, they improve as a population. Okay, if you are improve as an individual plant basis, the results are not that much good, especially like that's why we need a population improvement approaches. Okay, hybrid breeding, that's another case. But as of new synthetics, composites, population improvement, to take all the methods, they improve population as a whole because all the individuals should be maintained in the equilibrium. They should have random union of the gametes between the individuals of the population, within the individuals of the population, so that you should improve the entire population. If you improve only one population or one individual among the population, you are neglecting all the individuals. They are highly cross-pollinated. They definitely cross with other individual. In the next generation, your yield will be reduced. If this individual is not good, this individual is good. How will going to get improvement in the crop? That's why you should improve population as a whole. That's why it is called as population improvement. Okay, you should improve all the individual. You should bring all the positive alleles in this population so that even though it having 10 individuals, all the individuals should have all the positive alleles. Initially, very large population will be there, one having negative allele, one having positive allele, one having negative allele. But by selecting and aggregating them in a single population, they should have all the positive alleles for the desired trait so that you will get an improvement in the population. This is the one of the summary. This is the summary of entire population improvement. There are different schemes. We can improve for the GCA, you can improve for SCA. Either you can improve for both GCA and SCA and both population at a time in a recurrent scheme. Okay, reciprocal recurrent scheme. Okay, this is what the basic idea of recurrent selection. Then you can see the general scheme. There are different parents You're going to select and you can intercross, then again intercross, okay? Then you fix it, okay? Then you go for multiplication phenotyping and improve. See, you can bring, for example, it is having a good, having a QTL for one trait, it having QTL for another good phenotype, having different QTL. You can aggregate all the different QTLs, like it is called as QTL pyramiding also. That is another thing. If you're pyramiding into single individual, that is QTL pyramiding. You just should think it is a improvement of the entire population. Okay, with and if the population is having 10 individuals, they should contain all this. They all the individuals should contain different QTLs so that they can improve the phenotype. This is what the idea of recurrent selection. Then why we need of, I think I don't have that image. Then how we can through phenotypic selection, what we have to do? We normally rely on phenotypic selection. But for example, we need a separate seasons for the different selecting the different phenotypes. But you have a marker. For example, you are only relying on the phenotype. You have to select and you have to wait for another generation to cross, and again you have to improve. Then you have to go for another cycle. But if you have a marker, remember one point: if you're relying on a marker assisted selection, we can select and cross in a same time, same generation. Early generation you just select and you cross them, and you can do the two things in a single time. Means for every cycle you can reduce two generation. Two generation is clubbed to only one generation. Means selecting and self crossing occurs in the same generation. So that if the normal marker restricted recurrent selection takes 6 years, sorry, recurrent selection takes 6 years, your marker restricted recurrent selection takes only 3 years. Okay? Then how to do that? There are again several things are there, like should be based on how you will do, how you will select and how you will implement, how with we will calculate individual marker scores, like selection indices scores. I will deal when I was coming with the selection indices chapter. What is the score? How to calculate the selection indices score, marker selection indices score, then how to weigh them, how to use them in the marker recurrent selection. That I will deal in when I will. If you don't know what is selection index, how to calculate the selection index value, it is no use of telling now. Okay, but you just remember we can do selection and crossing in a single generation so that we can able to reduce the time so that we can improve the population at earlier period only. It takes very less time to improve the population when compared to your conventional marker restricted recurrent selection. Based only on the phenotype. If you integrate the marker, you can able to reduce the time, and we can able to calculate okay which QTL is present here. Take that only, that portion you can add into the population, and it is easy to screen also. If you are only on phenotypic, it again environmental effect will be there. You have to eliminate it again a tedious task. Yes, you should remember. Okay, what is marker recurrent selection? And another thing you should remember that selection indices, marker based selection indices. I will discuss when I after discussing selection indices chapter. But as of now, you just remember this. Then another scheme is called as marker assisted gene pyramiding. What is this? For example, you take this is a classical paper. Last in ASRB, I think 3 to 4 questions directly from the single paper. This paper is called as Joseph et al. 2004. Actually, now he is the HOD of Genetics Division IRA, Gopalakrishnan. You refer this paper. You just type marker assisted backcross breeding in rice by Gopalakrishnan S in 2004. It's a landmark paper. From there only, like I think this is another paper. I think Sama Masuria. There are XA21, XA31. Actually, this is another paper actually, but you just study that paper. which genes they have used. Okay, actually the blast blight is a bacterial blight is a very important disease in rice, and so many things are done to manage this disease, but nothing is worked. Ultimately, we have to use the host plant resistance to manage this disease. Now we'll study by taking this as a case study. We'll study what is marker restricted gene pyramiding. For example, they are the different donors. This is one of the cultivar like you take Pusa Basmati 1. It's highly susceptible to the bacterial blight. Pusa Basmati 1, it is highly susceptible to the bacterial blight. There are several donors which are identified like XA21, XA13, XA5, XA4, and they are from the different sources. I had to bring all the different genes governing for a single phenotype. What is the phenotype here?
Resistance to bacteria.
Yeah, resistance to bacterial blight. The pyramiding definition is remember. You have to bring the different genes from the different background into the single genetic background, which are governing for a single trait. If it is a multi-trait, again it is called as multi-trait introgression. It's not called as pyramiding. Pyramiding means all the different genes from the different donors coding for a single trait. That is only called as pyramiding. If all the genes coding for a different trait, it is called as multi-trait introgression. Multi-trait introgression.
Sir,
There is a
what is
difference
gene
between
stacking?
gene stacking is another thing. It is a deployment of the genes either through seasonally, either through spatially. Either it may be in the single background or in the different backgrounds also. It is just a method. It is a part of gene pyramiding also. For example, your rust. I think I will explain later. Okay. In another slide, I will explain. First, I will clear. Actually, it is
Sir,
either there are two types: spatial deployment and temporal deployment. That I will discuss. Okay. After discussing this, we'll come to that.
sir, here
Yeah.
we have to choose agronomically superior varieties of it or
No,
transfer.
nothing like that. Whatever the background it is. See, that is I told in back cross. What is the important thing you should remember in the back cross? This is back crossing only. But there is I discussed only from this one parent. If you are bringing the genes from the different parent into single background, but everything is same. This is back cross scheme only.
Okay, sir.
Just we have a different donors and all they are coding for a single phenotype. Okay. Rest other is same. There are different schemes again. In a backcross breeding, there are again scheme. There are tandem mating, symmetrical. Okay, there are different three schemes for individual backcross. That I will discuss. Wait. Okay. Then you are bringing a different genes governing for a single phenotype into single background. For example, you take Pusa Basmati 11 one. It is susceptible to blight and XA21. There are like IRBB. There are several varieties are there IRBB variety lines, donor lines. Then there are again a different method. How will transfer? Either you have to go single for XA21, you cross with recurrent parent, except like Pusa Basmati 1 and XA21, you develop a recurrent parent, improved recurrent parent in one backcross scheme for XA21. In another backcross scheme, TB11, sorry, I will use. Why the attendance is so low? See, there are different scheme actually. The mark gene pyramiding. One is one, two, three. Either you can go recurrent parent XA21, then you do recurrent parent improved by separate backers. For XA21 you do one backers and XA another one gene you do another second press. Then you can cross.
Stepwise transfer, sir.
Hmm?
Stepwise transfer, simultaneous transfer,
Yeah,
simultaneous but
that's
separate
thing. That's
transfer.
thing. I'm coming. Okay. Then you cross among them. Again you improve. Means how much time it will take. For each doing at least to take 3 to 4 years. For each backrest you have to spend 3 to 4 years. How much time it will take? That's why nobody is using. Like one. Another one is recurrent parent with XA21. Sorry, donor. Again back cross BC1. Again you cross with another donor. Then in BC2 again you cross with another donor. Until you cross with several donors, then when you select the plant which having all the four genes, then you start selfing. This is called as stepwise transfer. Okay, sorry, tandem transfer. Then another one is you can use first you cross in one cross you make recurrent parent donor parent F1, recurrent parent donor parent F1, recurrent parent donor parent F1. Then you cross them. Then finally you will get some F1 plant with all the genes. Then you do backcross. Okay. I will show that. I think.
Whether this particular locus is single locus.
No. Why it is coming? I will show the image actually. I didn't kept here. I will show the image from the book. Don't worry. Wait, I'm opening the book actually. Hmm. See, what are the notes I'm preparing from this book only? Okay, you read this book. And even some points I will going to miss here. You just don't think that it will not important. Okay. Um, I'm giving a roadmap so that based on that you should plan your study. Okay. See, here there is a separate background. First you cross with recurrent parent with donor. Its slides is visible? No? This image is visible?
Yes, sir. Yes, sir.
Yes, sir.
Recurrent parent and donor parent F1. Recurrent parent, you again create a BC1 F1. Again a recurrent parent means a separate backcross. As I told, See, for one gene, there is another one backcross. Another donor parent, another backcross. Another donor parent, another backcross. Another donor, another backcross. You know, at least you take 4 years for each backcross. How much year you have to spend? How much time you have to spend? Then again you cross recurrent parent into recurrent parent, recurrent parent, improved. Then you create F1. Then you cross them. Then you keep on selfing and you fix it. This is what a separate backcross scheme. Then another scheme is called as symmetrical mating, and another one is called as a tandem mating. In this one, you can see in a symmetrical mating, what will happen? Recurrent parent, donor parent will creating in F1, recurrent parent, donor parent to F1, and you cross this F1, and again you will get double cross. So that you have to create double cross for another two different parent. Then you cross it. Then again you come to this one F1. Then you start keep on doing backcross scheme. Means one backcross scheme with four parents. Okay, this is called a symmetrical mating. Then another one is called tandem. Means just DP1, DP2 keep on crossing the donor parents. Then you start backgrounds. Okay? This simple image you just go through. You will go into you just observe the things clearly. It will going to understand. It's nothing like any explanatory things are there. Okay, understood this what is symmetrical mating, tandem mating, and the separate backcross program for the gene pyramiding. Have understood or not?
These are in molecular breeding book, sir, AKC book.
Yeah, this is I directly took from book on. Okay, the examples, everything, but not images. Okay, based on the citations, like they will cite now. You just cite that paper, then you download the paper images. I got images like this.
Sir,
But these
the
are the scheme is there
scheme
in the
you,
book. There in
sir,
the book.
these schemes are for a single trader, sir.
Yeah, they are from they are for single trait. This is how
Okay.
differently you can able to transfer the different donor genes from different donor parent into single background. There should be some scheme na how to cross. Either they cross either this pattern, either they cross this pattern, either this pattern they can use, either this pattern they can use, either this pattern they can use. Okay, there are several mainly
Sir,
time. It's based on the ease of the breeder. They can choose the mating systems.
sir,
Okay.
I'm fine. I don't know, sir, whether it's a doubt, sir, because there are four donor parents, sir.
Yeah.
So they will have different types of alleles for that particular trait, sir. And if the
What?
trait is, they will have different types of
Different?
genes, sir, for that trait, isn't it, sir? Different genes, sir. Donor
Yeah,
parent
yeah,
one
yeah.
will have
See,
one
I
gene,
showed na
donor
in
parent
image.
will do. Yes, sir. So
See,
there
in
are
this
only
episode,
two alleles for a monogene.
I told
Sir, okay, sir. There are only
no.
two alleles, sir, for that particular
Who told?
locus.
Only two.
No, sir. If we are going to
I'm not
fix, how
talking
can you?
about locus here.
Yes, sir. We are going to fix,
See,
how can we fix all
if
those
based
four
on the
alleles,
marker
sir?
you select homozygous cell. Yeah, that's what the markers you are using now. You are using markers now. If you are fixing marker dominant allele, then that's why when you are getting homozygous condition only, you just selecting that plant only. For
Yes,
example,
sir.
for this
Understood, sir.
gene, either you have to check either the gene is in heterozygous condition or in homozygous condition. For example, in this particular plant, you take these are the progenies now as of now. You just forget it. Okay, for example, this is the only progeny. You have to check this progeny for all the genes, either they are homozygous condition or not. For XA21, it should be homozygous. XA13, it should be homozygous. XA5, it is homozygous. XA4, it is homozygous. Okay, then you further go. That's why if you don't have marker also in conventional system, how are you going to know? You just advance it. F2 derived, for example, if it is in F2, you just advance it, just self it. If it is segregate again, if it is some segregate again, you just reject that plant. What we will do that only now? It's a common procedure. You want to check whether this gene is homozygous or not. That's what you have to do. You have to analyze all the plants. Which plant is having the gene which are both are in the homozygous condition. Because all the genes are in the homozygous condition and it's exactly similar to the recurrent parent. Okay. If you read on paper, Khanna et al., I think in 2016, I think, or by Ranjit Kumar Ellur, one paper is there by Dr. Ranjit Kumar Ellur, my MSc guide. Excellent paper. It published in the application of genetics for the blast. They used 8 parent, I think 8 PA genes into single background. You just read that paper. You'll get to know. Understood this one? What is pyramiding, gene pyramiding, these things?
Yes.
Keep on reading the things, papers. These are the trending thing. Like there's so many paper every day we get one paper on gene pyramid. Like when we finally come to the what are the advantages again, like phenotypic screening is very expensive and sometimes difficult and impossible. Like you take male sterility, sometimes it's very difficult to screen. When the trait having a very low heritability, that also conventional selection will not work properly. When the selected trait expressed late in the plant development, for example, you take fruit antioxidant content. Okay, when at ripening stage only it will get difficulty. Biochemical traits very difficult to analyze. That too when maturity state. That time how we'll do the selection. So that's marker sheet came to play role. Then incorporating genes for resistance. For example, if you want to incorporate different genes into different background, through only phenotypic selection, how we'll get to know? All are bacterial blight resistance only. Either this is due to XA21, or this is due to XA13, or due to XA5, or due to XA4. How we'll get to know? If you don't have any markers. That's why gene pyramiding is a very, very difficult task. Can't imagine also without the use of markers. Then when expression of the trait target gene is recessive. You know, when the gene is for example, resistance is gone by recessive. We need an extra year to self it and you have to see the expression of the recessive homozygous. It will doubles your backcross breeding time. But if they're using marker, it's not at all depend on the genotype of your trait. It just depend on the marker genotype. There is no need to modify your any background scheme for the recessive trait or any trait. Just the standard method you can use. That is the greatest advantage of marker restricted selection over the conventional selection. Then we can able to accumulate multiple genes which are governing multi-traits also. That is called as multi-trait introgression. Multi-trait introgression. For example, if in a single background, we can bring high yielding and highly resistant to blast, blight, to single blast, blight, nematode resistance, whatever it may be in a single genotype. That is called as multi-trait introgressions. Then these are the different schemas we discussed. No need to go. Okay, then genomic selection. I will discuss after the genomics. Now I think you are clear with what is marker restricted selection, what is marker restricted backcross breeding, what is marker restricted recurrent selection. Then he asked about gene deployment. Here, it is there. Marker gene deployment. What is gene deployment? Means, for example, think it is India map. Sorry, any map. This is Karnataka map. Think like in one district, here there will be some disease will be there, and through air it will coming here, and it will pass here, and it will pass finally here. For example, from Nilagiri to that some rust, the teliospores will pass like this. Now, if you deploy some genes here, and deploy some genes here, some deploy some genes either temporally, either season wise or spatial wise, so that it restricts your disease development. That is what gene deployment. There are several types. Okay. But your gene pyramiding, you are not doing all those things. You're just bringing the different genes into a single background. That is the only, and it's going for a single trait. Here, you're planning the release of, for example, you release the variety which having uh, one resistant gene so that it can block here the spore spreading. So that is the word deployment. You are deploying or you are putting the cultivars which having the different resistant genes so that your resistance is break either into single background, how you are utilizing your resistant cultivar or the plan of utilization of the resistant genes either through the time or through the space. It is called as gene deployment. Gene pyramiding may part of gene deployment. Okay, but the gene deployment is the scheme. Now you understood, I think.
Sir, gene deployment and gene stacking same one, sir?
Yeah, stacking. Yeah, that is another thing. Stacking is there, both are mostly same, but this deployment is a broader term. Stacking means in a single variety or a different variety, you keep on increasing your resistancy. For example, in at one stage, for example, you stacked some three resistances, but again you started epidemic. You stack the different genes, so that temporally and special again you're stacking the gene so that we can um, uh, break, we can create a barrier to stop the epidemics of some disease. Okay,
Sir, in multi-lines we follow this, okay?
that also there are so many methods. Okay, but you just clear with the word what is gene deployment, stacking. Stacking, they are all interchangeably we are using. Okay, stacking is the one thing, it's a one strategy of gene deployment, you think. Okay, gene stacking is also one strategy of the gene deployment. And gene pyramiding and bringing all the gene multiple, use these varieties accordingly. This is also one type of gene deployment. Okay.
Sir, sir, in stacking, we are inducing the genes in one variety.
Maybe sometimes it may be in different donor, different varieties also, and using them in a single time. That also possible.
Yes.
Sir, then
We're stacking
then
in that area. We are putting all the resistant genes. That also comes. Either I had to check.
then sir, multi-trait integration and this stacking looks little bit similar.
Same. Okay. Sometimes it may same. Okay, that's why there are different concepts. There are different strategies of uh, disease management. Okay, through host plant resistance. I will deal these things in a separate class. Okay, this is the different strategies. There are several different are there. Maybe they can use even the varieties which are developed through gene pyramiding. They can be deployed according to different space, according to the different epidemics of the disease. That is one of the deployment strategy. And stacking also, you stack these improved lines
Sir, audio.
Sir, your audio is not.
Sir, you are not audible.
gene deployment, deploying different genes on. Those things I will deal. Disease resistance management, I have separate classes for that. Don't worry. Okay. But as of now, you
Sir, your
understood what is
voice.
MABB, what is MARS, what is this one. Hello?
Hello, sir.
Yeah.
Sir, your voice was cut in between.
My voice. Okay. What is your doubt? See, I will deal all the disease resistant management strategies develop what is deployment, what is the types of deployment, what is stacking, okay, how they are, like what is different types of deployments with examples. I will deal actually. Okay. Some example I'm
Instance
unable to
readings.
recall, so that I'm not able to clearly able to discuss. But this is the only difference. Okay. Even gene pyramiding may be the strategy. You can use the improved varieties to develop gene pyramiding and deploying at different time and different space. That also can. There are different strategies of managing the disease epidemics. Okay. But marker sheet selection schemes, you are clear. All the schemes, genomic selection, I will discuss after discussing genomics. All these schemes are clear except genomic selection. This
Yes,
early
sir.
generation mass is nothing but you are selecting at early generation and you are advancing easily. I think all of you clear. I think only 4 students are there today. I don't know they are getting molecular breeding or not. But tomorrow I will deal, I think, recombinant DNA technology or genomics. Actually, these are the examples are there, case studies I directly kept. We can go with this papers. Actually, this paper is also very important. It's a landmark papers actually. Okay. Then genomic selection I will discuss. I think I have some questions. I think, yeah. See the genes for bacterial blight resistance.
XA 13 and 21.
Yeah. See, these are the very important things. Another, this is genomic selection. In this year they asked reliability of the selection only. Then they asked like nils and see nils, DH and rel and F2. Which are the population are heterogeneous here? Heterogeneous background.
F2, sir.
Sir, if we consider more double applies, it is also heterogeneous. Will is
Yeah,
also heterogeneous. F2 is also heterogeneous.
yeah. This is the thing. Okay. Except nil, I'm asking heterogeneous background. If you have you take real population, all the individuals are same. Then I told all the class which I made it is like useless now. How I told more is the recombination, more you was helping, more you will getting resolution. What are the things which I discussed? It's a waste of thing. Where homozygous means even the recombination happens, what will happen?
Heterogeneity will maintain, sir.
Heterogeneous background. Each
Yes, sir.
rel is all the rel are there different from one another.
Different.
All the DH lines they are different from one another. But at particular locus they are homozygous in nature. But this individual is different from this individual. This individual is different from this individual. Again, you just make them cross. Again, you'll next generation you will get into recombination. Like this will happen. That's what. Okay. They are except nils. Nils is they having same genetic background. They are differing only with the several only one or two traits, one or two genes. That's why while fine mapping, we have to cross the nils, two different nils. Then we are creating an F2 there. Because if you are using nils, we cannot get this one, like high recombination. That's why we are crossing homos two different nils. That's what you have to understand. Okay? Think you got this point. Then another question there. This question is asked in the exam. So many people have been. So I discussed these things. See, these are a simple logic, but people will not apply the logic. They just blindly mug up the things which are, and some people are some like very dangerous. Like they're beheading the points which are present in the slides. And if the question is not from the slide, then what we can do? That's why you should have an analytical. We have discussed everything. When you read the books, when you read more, you'll get like your thinking power will be more, so that you can able to apply and analyze the things in the exam. Then another question was nils, how we can develop the nils? Through mutation, through gene substitution, through transgenics, either through backcross. All method you can develop nils. This is the question they were asked. They were asked A, B, C, D, only A, B, C, B, C, B, only A. They were asked. I think all are. Through mutation you can create nils. Through gene substitution you can create nils. Through transgenic you can create nils. Either through backcross you can create the nils. All four methods are possible. These are simple things. You should apply your brain there. To apply that, you should clearly know what is nil. Okay, okay, bye. That's all for today. Anything needed to ask?
Sir, mutation, transgenics, backcrossing, and others. What?
Gene substitution.
Okay, sir.
Like one of the method is chromosomal segment substitution lines. Through that method only, through that also you can develop nails.
I forgot to ask
substitute.
yesterday, sir. Chromosomal segment substitution lines also can be generated, or foreground selection and background selection can be used to generate CCSLs, sir.
Yeah, you can, because now if you want to have a donor parent this genome, you can select that part only, that line only. That also can be done.
Sir, generally through which method they will do, sir? CCS, CSL.
MABB only. MABB nowadays we are using MABB. Okay.
Okay.
But there is no foreground selection, are there?
Yes, sir.
Only
Only
how
background
much
selection.
recurrent
Yes,
parent
sir.
genome?
Only
That is,
background.
we are not having any particular gene has to transfer. Okay. It's like uh, how much recurrent parent is, how much in one line, how much is recurrent parent and how much portion is donor parent. That is the only important thing. The donor parent segment may be your foreground selection, and rest other is background selection. I think no recombinant selection. Okay. So many points. You keep on reading MAS book like marker selection book by Akising and BDC. There's so many doubts will come. Again, you read. Okay, you can search in the Google. I can read some papers. Then this is a trial and error method you should follow for molecular breeding. Not like that single time listening class you can get all the things. It's like keep on reading, keep on practicing, then only it will come. Okay, that's all for today. I'm ending the class. Good night. Good night,
Thank you.
everyone.
Good night, sir.
Bye bye. Thank you.
Thank you, sir.
Thank you. Thank you, everyone. Bye.